Should be able to hear me now. Does it work well? Okay. Well, thank you very much for the kind introduction and the warm words. I'm delighted to give this inaugural lecture. As you mentioned, I arrived at LSTM not even 2 years ago, and it feels really good to now having the chance to share a few data, but maybe more important, my previous experience, my path so far, and maybe also some advice. I know that there's many early career researchers here, so I think it's maybe also interesting to share what kind of approach I have in different things. So the title is Hijack, Persist, Evade. So I think this very nicely recapitulates how the most dangerous, the most problematic human pathogenic viruses behave. And I decided to devote today's lectures to a large part to a virus which really keeps fascinating me, which is also the first research topic I started working on when I started in academic research. So starting off from a really deadly disease, an incurable and still incurable disease, and HIV-1 has now turned into a manageable chronic disease. And I would also like to mention that I'm, of course, a researcher working on HIV-1, and we always tend to really emphasize the importance of our research topics, but of course, I'm very well aware that HIV-1 is one of the important pathogens, but there are many more. However, I'm a virologist, so I like to work on viruses. So I will start with giving a little bit of introduction about the problem, and then give some insights into my career so far, the different steps I made, and maybe stop with some outlook. So a few numbers. In 2023, this is the distribution of HIV-1/AIDS we have globally, and we still have yearly a number of 1.3 million individuals acquiring this infection. And these numbers are actually stagnating a bit. So it's not very much, there's not really a trend to see that these numbers would have been reduced even further. We have almost 40 million people on this planet living with HIV, and this includes those who are aware of their status, but also those who don't. And HIV-1/AIDS is one of the few examples in the virological field where we have really a fantastic array of therapeutic possibilities. So we have a large group of different classes of drugs which are antiviral. And if you compare the number of medication people had to use on a daily basis, this has really very much improved. We now even have prophylactic treatment, which is offered to persons being at risk of acquiring the infection. And this does not only protect every individual to acquire the infection, but it also helps to really reduce the global spread of HIV-1. We also now have the principle U equals U. I hope you're all familiar with this concept. It means undetectable equals untransmittable, which means that if a person which is positive for HIV-1 is aviremic because they follow the treatment and are below detection, these persons cannot sexually transmit the virus, which has made a really big change for people affected. And we have really exciting developments, again, on the therapeutic side. So we now have long-acting drugs here, a capsid inhibitor, which was, I think, rightly described as a breakthrough of the year, lenacapavir, and the studies are just amazing. They showed in some studies up to 100% efficiency against acquiring the infection if you take this drug prophylactically. And the interesting aspect is that this is a drug you need to inject every 6 months only. So this kind of drug can really make a difference. So that's quite exciting and promising, right? And I even got in touch with people who told me, well, maybe you should start looking for another research topic because I think HIV-1 this problem is being solved. And there was really an ambition to use test and treat as the major strategy to really reduce the curve. And we had, of course, different voices in the politics and in the research field being very confident. So Donald Trump said 2019, we are stopping HIV-1/AIDS and its tracks. It's pretty much stopped. And even Mike Ryan was a bit more careful, but still made the statement, HIV has not gone away, but we have come to terms with the virus. People don't feel as scared as they were before. And indeed, the numbers looked promising. So there was a trend to reducing the overall numbers of HIV-1 acquisition. But I'm sure you all know the recent developments of many nations, some nations deciding to either pause or interrupt or even terminate their global health programs, which were actually providing test and treatment access to communities affected. So there's now some really interesting modeling and projecting studies coming out, which predict in the worst case, we will have between 40 million new HIV-1 infections. But coming on top of those, we would anyway have predicted in the next 5 years. So these modeling, of course, there's different scenarios depending on whether these global health programs are really stopped forever or just interrupted for a certain amount of time or reduced. So we don't know. And the numbers regarding the deaths related to HIV-1 showed a similar trend. I mean, again, we did not reach the target of UNAIDS 2025. But we were close to actually reach those. The projections now actually look very daunting. So we should be prepared to a significant additional number of HIV-1 deaths if there's no intervention. So why actually does HIV-1 come back when you stop to treat? This also is very plausible when you look at the individual person. So I really like to show this graph because it nicely shows after acquisition of HIV-1, the viral load is really shooting up. If you have access to antiretroviral treatment, the viral load will very quickly go down if everything goes well. But if this person goes on holiday and somehow the luggage with the medication gets lost, this person after 2 or 3 weeks probably will experience a rebound of the viremia, which allows the virus to spread even further. And basically the health problems would really start immediately. So this is just to show you how vulnerable this strategy is to really rely only on medication, although this is a tremendous success. But it's a very fragile construct. We really depend on constant and secure access to antiretroviral treatment to keep the viral load low. So maybe I didn't mention this. This is a lifelong treatment required. So there's no cure and no HIV-1 vaccine. And I think it's fair to say that this antiretroviral treatment is great, but it's unlikely that we will really eradicate HIV-1/AIDS as a global problem simply relying on giving access to treatment, because, as I already mentioned, this is very vulnerable to political goodwill and decisions. So I will now try to explain to you a bit at the molecular level what makes HIV-1 so special and why it's so much more difficult to design strategies against this virus as compared to other viruses. I prepared a little cartoon to show you how the virus is basically infecting cells. We start the replication cycle with a virus particle, and when I use the first word of my title, will hijack components of doesn't work. Yeah. So the virus needs to exploit or hijack cellular factors in order to be able to infect cells and to propagate in these cells. So the first step is to interact with the main receptor, CD4, and a co-receptor, which is this yellow guy here. This is very, very essential and basically only works in the human system, which explains why other animal species are not susceptible. They cannot be infected by HIV. The next step is then that this interaction will induce the membrane fusion, where the virus delivers its inner content into the cell, followed by squeezing through the nuclear pores. So the virus is transferring its genetic material into the nucleus of an infected cell. And then, which is very special about HIV-1, is it integrates its genetic information into our cellular chromosomes, which is quite a clever strategy from the virus perspective, because it enables the virus to be persistent. So it's not a transient infection, it really fixes its information for the lifetime of the cell. And this explains why HIV-1 is a persistent and chronic infection. Which will become a bit more important later is that HIV-1 not only does this, what I just described, these first steps also happen in total silence. The T cell is unable to really sense and react to these events. another virus would really trigger some kind of alarm machines. The cells would ramp up antiviral measures and start fighting against the infection. In the case of HIV-1 has really evolved to sneak in like a Trojan horse, and this cell is not really making much notice of it, just minding its own business. So after integration, this, what we call the provirus, the genetic information, which is now part of the cellular chromosome, will basically behave like a cellular human gene. It will be expressed. We will have proteins being made. They assemble at the plasma membrane. And you see how new viruses are being built after all the components have been put together. And the virus will then be released. Of course, these new virus particles would like to find new target cells to infect another cell further. So this might happen. So that's the journey of HIV-1. This is how it accomplishes its so-called replication cycle. And actually, if the story would end here, our immune system might have a chance to get rid of, because when the virus expresses its proteins, that means the cell which is doing so is visible to our immune system. So we have a lot of mechanisms in our immune system that could then target these cells specifically and eliminate them. And then maybe it would just be an acute infection, like we know from some other RNA viruses as well. But HIV-1 has another trick. It goes latent in a proportion of the cells it infects. So this shown here on the bottom right part. Not all the viruses result in active replication and active virus production. A part of them will just go sleeping. It's like a sleeper protocol. You have an inactive version of the viral genetic information, which is just waiting for a good opportunity to be waken up again and restart. But while the virus is latent, so inactive, it's invisible to the immune system, which means that the immune system has no chance to target it. And this is the real problem we have with HIV-1 and why it's so difficult to design cure and vaccine strategies. So I started working on HIV when I started my PhD, and I did so when I met Oliver Kepler, who was my very first supervisor in academia. So Oliver just returned from his postdoc in San Francisco, which he performed at the Gladstone Institute, and he was looking for his first PhD student. So he was a medic, and at the early 2000s, San Francisco was clearly one of the hotspots of the epidemic. And he was also looking after patients, and he told us sometimes some really gruesome stories about patients suffering from this disease. This is how I got actually exposed to the entire topic. But at the very bottom of his heart, I think he was really an experimental virologist. So he really loved to look into mechanisms and understanding the biology of viruses further. So the project I was interviewing for was about the development of a small animal model of HIV-1, actually quite translational topic, if I think back. I already mentioned other species are very hard, they're unable to be infected. So we can use non-human primates to a certain extent, but you can also only use related lentiviruses, not HIV-1 exactly. And of course, we have ethical concerns. Nowadays, studies use humanized mice, so mice which are transplanted with some human tissue or human cells, where you can do a limited set of experiments. But we were interested in genetically modifying wild-type small animals and to adapt them and develop them further to become susceptible to HIV-1 infection. This would be really necessary to do large-scale preclinical studies, whether it's drug testing, but also vaccine and curing. We really wanted to have a species or an animal model which is immunocompetent, which the humanized mice are not. So in this PhD, I enjoyed the supervision by Oliver. He was very hands-on. And if any one of you is considering starting a PhD with someone who is very fresh in the system and maybe just finished his own or her own postdoc, I think it's a wonderful opportunity. So I learned extremely many things. We worked together at the bench, and I learned so many things which I still need today. So I'm very grateful for this support I got. What did we do? We generated rats which express human CD4 and human CCR5. And these animals were infected, and they showed sign of infection indeed. So showing that these transgenic expression of these two factors was relieving the first block, but the viremia was quite transient and quite low. So I then looked into what could be potential reasons and how could we advance this further. And I looked into rat and also mouse CD4 T cells, where I studied which steps the virus is able to complete in these rat cells expressing human CD4 and CCL5. And we found that, well, actually in rat CD4 T cells, everything is working pretty well until integration. And we even saw some early viral gene expression, but then the virus gets stuck. There must be existing other blocks, and these non-human species would prevent the virus to really finish this replication cycle. So nevertheless, we got in touch with GSK back then and tested in a preclinical setting a GSK, an integrase inhibitor, preclinical integrase inhibitor, which was really exciting because basically we used this model for what we actually intended to use it, even though it was not fully finished. So I graduated in 2007, and Oliver was not very keen to terminate this quite productive scientific relationship we had. And I was a bit hesitant, but then I accepted when he promised I would be allowed to shift gears a little bit. So I decided to stay for 3 more years, and I was lucky enough to having access to 2 postdoc fellowships, which supported me during this time. And a very hot and interesting topic back then was the analysis of so-called restriction factors. So we all have in our cells antiviral factors, which block virus infection or the spread in our bodies. Kind of, yeah, natural defense mechanisms we all have in our cells. And I'm going to introduce one very specific one we worked on because it's nice to explain, it's easy to explain. So normally, if you think about the perfect scenario from the virus perspective, as I explained, viruses are being made, they're being released, and these virus particles will then be diffused or they will be transported in the bloodstream and find the next cell they want to infect. So when you do electron microscopy of cell cultures where you did an infection and then you want to mimic the whole thing, you have actually a hard time to find virus particles because they are all washed away in the procedure. However, if this red guy here is being expressed, which is one of these antiviral factors we express in our cells, the viruses would basically stick to each other and they would stick to the plasma membrane of the cell they actually want to leave. So this prevents these virus particles to leave the cell and infect new ones, right? And this is quite impressively also visible in these electron micrographs, where basically human tethering, so it was named because of its function, its ability to tether virus particles to each other, it really acts like a molecular glue. This is, yeah, quite simple factor to explain. So now you're probably asking yourselves, well, but she just said it's protecting us from infection, so why actually is HIV-1 then a problem if we have these potent antiviral factors? Well, HIV-1 is one of the smart viruses, and HIV-1 has evolved to counteract these immune defenses. It expresses one specific factor, this blue guy, VPU, and the job of VPU is to downregulate tethering from the cell surface, so to take it away from the point of action and lead it to degradation, which basically means in the context of HIV-1 infection, the virus is investing some effort to get rid of this very simple restriction factor, which is quite sophisticated and which explains why HIV-1 infection is so productive. And there are many more examples. If you want another layer of complexity, you think back to the rat model, where we did not see productive infection. We found that the mouse and the rat version of this rat tethering are resistant to this counteraction mechanism. So even if VPU is there, the virus would still be inhibited by tethering. This is why we don't see high viremia in these transgenic rodents. Okay, so another 2.5 years of we have passed. I was still in Heidelberg, and I thought, now it's really time to move on. Before I moved on, I delivered on one very important and very challenging project, which will remain the most important and the most challenging project in my life, probably. And I then started in Ulm as a very ambitious and maybe also naive group leader. So Frank is actually a very recognized, very highly awarded scientist specializing on antiviral immunity against HIV-1. So exactly the field that really interested me. He was highly decorated with different prizes, very well established. And somehow he saw some potential in me. He told me, yeah, Christine, you can start. I give you funding for your position in a technician for 3 years. And when I spoke to him, I thought he was trying to convey to me, well, swim or sink, but here, 3 years, you can do whatever you like. And well, Oliver was constantly tiptoeing around us in the lab in Heidelberg. Frank was exactly the opposite, I think. He was very hands-off, so I was entirely on my own, except, of course, accessing the infrastructure. So I learned to write my own grants, manuscripts really on my own, to develop genuinely my own idea, to hire people and make a lot of mistakes. So it was a really a good learning experience. So I still was interested in cell-intrinsic innate immunity. We now looked at another factor, which I will just very quickly explain, because it touches another step. So I explained that when viruses are being made, you will have these spikes on the surface, and they act as docking stations to interact with the receptor and co-receptor during infection. However, if we overexpress this, or we have this factor in excess, we basically found that the viruses which were made were almost naked. So they had very few spikes only, and this resulted in a very low infectivity. Two data for those who are interested. So we knocked down anti-K, which means you modify the cells that they no longer express this factor. And this resulted in a 10-fold higher infectivity of the virus and also a much higher abundance of these green spikes. So it makes a lot of sense. So why is it important? We could use this strategy and develop an antiviral mechanism or drug that would basically mimic what 90K is doing. And we still have some ideas about this and think it's wrong. I think it's definitely worth pursuing. So this was the time when I secured my own first research grant as PI in own right. Would like to stress only in the second attempt. So don't panic if this doesn't work on the first one. I remember I was really shattered when I heard about the bad news when my very first grant was rejected, but just for a couple of hours. I went for a walk and then I went back to the desk. And it was, these decisions take so much time from submission to the decision letter. In these 6 or 8 months, so many more data had already accumulated that I basically resubmitted with more data, and then it went through. So this is a very normal experience. Typically in these kind of lectures, we only talk about our successes, and not so much about all the failures, but they are very real. So remember, the clock was ticking. I had 3 years, so I was quite happy when at the end of the 3rd year, I got selected for a 5-year habilitation fellowship of the state of, what's in English, Baden-Württemberg. And this fellowship was supposed to basically allow me to acquire all the qualifications needed to become a professor at some time in the German system. However, this letter of acceptance into this program coincided with an offer I got from Hannover Medical School. And I found this a bit more attractive because this was the first step in the academic ladder, so the assistant professorship, which was actually coming with a seducing package as well. In addition, Hanover was much more close to Hamburg, where my husband, his family, my parents were based, so everything fit. So I decided to give back this habilitation thing, and because this was actually tied to the specific state in Germany, and I went to Hanover, to the Twin Core in the Institute of Experimental Virology, headed by Thomas Pietschmann. It's still headed by Thomas. So Thomas is an expert in the HCV field and is also working on other RNA viruses. So this was the time where HCV direct antivirals were really showing success. So I got confident to to open or to widen our research portfolio. We started our baby steps into chikungunya virus and other alpha viruses, starting to establish tools and did work in collaboration, but also our own. Nevertheless, we still, of course, were continuing our HIV-1 research. Again, I got a bit distracted and was now more interested in sensing aspects of the infection. So how do cells recognize that they get infected and how do they react? or how do they not recognize actually they get infected by HIV-1, and how does this all work? So I'm not going to explain this slide. I think this is probably not required. What I want to really convey here is that we identified that infected cells, infected T cells, communicate with neighboring monocytes or macrophages if they have a chance to sit together, like you think could happen in complex tissues like lymph nodes. And normally, if you infect T cells with HIV-1, as I mentioned at the very beginning, T cells do not react. They're not aware of the invading infection. But they can communicate to the neighboring macrophages and basically finalize a signaling pathway much more quicker, much more efficient. So basically, different cell types talk to each other by exchanging information and alerting each other about the invading infection by having these intracellular contacts. So this study took eternally. So we worked on this really, really long time. And I got really pressurized from so many people telling me, you have to publish this now. You're working too long. You have to finalize this. You have to deliver. And I was quite stubborn. And I want to publish when I think this is complete. And I like the entire study. And I was successful. I think I was very lucky as well. Could have gone wrong. But we were successful in publishing this study in Cell Host and Microbe, which is a quite well-received journal. And this was really our ticket to move on. So we got many awards. This was well-received in the field. And I think it was, for me, a golden ticket to the next step of my career. First of all, why is this actually important? I try to remember to ask this question. It's quite fundamental research here. But now we know how cells communicate to each other and how this potentiates immune responses. So the little substance, which is actually playing a major role here, has now been added as an adjuvant to vaccines because it can easily help to boost immune responses. So it is very fundamental, but it has a connection to translation. So I was now a couple of years in Hanover, and tenure was looming, which means that there were some discussions, but everything was quite lengthy, and I was not exactly sure what was going on. And I got an offer from Charité in Berlin, because I like to work on different options, typically. And this was 2019, and I joined the Institute of Virology. 2019, we established the team there. And then the pandemics kicked in, and I found myself sitting next to Christian Drosten. Maybe some of you in the audience know it. He's the international go-to person if you have a question about SARS-CoV-1 or MERS-CoV. So he was a really well-renowned coronavirus expert, which was not so important for me when I accepted, but suddenly was really very, very interesting, because we were then really embraced in this institute and were able to conduct totally new projects by merging the innate immune expertise that my team had with their very, very well-developed coronavirus expertise. We also gained some speed with our chikungunya virus research. We conducted the first more in-depth study doing single-cell RNA sequencing of primary synovial fibroblasts and managed to publish this. We looked into anti-interferon autoantibodies. I find myself in calls with clinicians fighting for authorships, sometimes successful, sometimes not, doing some really very clinical stuff, basically. So autoantibodies against interferon are one very important cause why elderly male were much more affected by the first wave of SARS-CoV, because you find these autoantibodies at quite high levels in this population. And this was also the time where I got friends with Daniela Niemeyer, and she is an expert in Christian's team, and we managed to conduct together quite fancy studies, again, as I said, by merging our expertise. But I wanted to talk predominantly about HIV-1, and in the shadow of the pandemics, we of course continued our activities. In this PNAS paper, again, you might think this question is maybe trivial, but in Cold Spring Harbor retroviruses meetings, this question would really fill entire sessions for hours. So we really conducted a study with all the controls you could think of to really show that T cells just do not perceive infection by HIV-1 as opposed to other viruses. And again, you might ask, who cares? There's no translational next step here in this particular study. But I think it's very important to also find out about the limitations of what can be done. And there have been some studies suggesting that we could exploit some natural sensing mechanisms to fight against HIV-1. And I'm convinced we show that this angle would not work here because the virus is just too far developed and is managing to infect T cells in complete silence. Yeah. Again, tenure was really so. If you want to get rid of me, you just offer me still stamps. And this is probably how this looks like here. Again, I had handed in all my documents, but I'm passing by. And then there was an October, a rainy September, October afternoon where this job advert flew into my timeline on Twitter back then. LSTM, new job alert, an exciting leadership role available at LSTM, chair of molecular virology, working closely with the head of department to undertake research in molecular virology. So I submitted my application. I thought I would never hear back from them, but you know the story, I'm here. So I had a farewell party in Berlin in summer 2023, where I said goodbye to this amazing team. Luckily, this was just temporary because they joined me 6 months later. So you see them here in social space a few months later. So that's the team. That's how the team looks like now. And here are several reminders I am trying to remind myself. First of all, Saskia, where are you? Happy birthday. Nice to have you here at your birthday. So this is basically depicting the current team. Amazing people. That's the second reminder for me is I really want to thank you. I'm blessed to work with you, and I really enjoy the conversations I'm having with you. These one-to-one meetings or group meetings are really the best and the most pleasant part of it. So please keep going. And no, I forgot the third. Now, the third one is I don't have time, of course, to mention all the other group members we've had in the past, those in Berlin and the other institutions. But I would like to equally thank all of them, as some of them are listening from online. And of course, you are all equally important to the successes we have. So thanks for that. Since I joined, I really felt very welcomed. Especially scientifically. So we were really able to make first connections. Some of these plans are quite advanced already, and I will now go into detail on all of them. In some aspects, it's just ideas, conversations, joint interest, but this is just to illustrate that I really feel I had a really good start scientifically, and there's so much more to come. What is our very own ongoing work? I mean, again, of course, we would like to continue on the work we were doing before, but as Giancarlo mentioned, and I think it was Hillary who explained it a couple of weeks earlier so nicely, other institutions want to fill knowledge gaps, and LSTM wants to solve problems. Sometimes it's not mutually exclusive, but I think here I have really the opportunity to develop more in the other direction. Or in the further direction. So we still want to understand how human pathogenic viruses work. We want to understand how and where we can attack them. We have now a real clinical problem as underlying question. We work now on HIV-1 cure. So this goes much more beyond understanding how HIV-1 infects cells, but we really want to advance this still very important gap. We still work on chikungunya viruses. We were quite lucky to join a EU consortium recently that will empower us to do some genetic screens to identify cellular factors which either inhibit or facilitate infection by these viruses. We characterize respiratory infections, SARS-CoV-2, and in the recent future, probably also influenza A virus. And we still are keen to continue our work on monkeypox virus. But since I promised to dedicate today's slides predominantly to HIV-1, I would like to introduce you on, not the very last slide, but probably the last scientific slide, how we would like to advance here. And first I have to make you familiar with the concept of shock and kill, which is probably the most advanced concept regarding HIV-1 cure. It doesn't work in the clinics, so maybe it will turn out it doesn't work at all, but it's the most advanced concept. So I think I already explained to you that the problem we have when we want to develop cure is that HIV-1 goes sleeping. So you have an inactive proviral DNA illustrated, shown here in red, which is not transcriptionally active. It does not make proteins. The cell just looks like any other CD4 T cell for our immune system. So to wake up the virus, the idea is to treat cells or people living with HIV-1 with so-called latency reverting agents. So their job would be to wake up the virus, to force the virus to go into the active mode, which would result in this green cell here. So mRNAs would be made, proteins would be made, they would be visible on the cell surface, and the immune system can now attack these cells and specifically eradicate them. And then you would hope, I just said this, eradicate them either by the immune system, or maybe there's even another factor which could be added. Of course, you have to include in this strategy or into this regimen antiretroviral therapy, because otherwise all the newly produced viruses, whose production you actually force, would of course infect new cells. So of course it's a no-brainer. It's very important to include ART. And the cells would then be eliminated. And ideally, you would have a situation where the patient would be cured. So this has been tried in many different clinical trials where people living with HIV-1 have been given different sorts of LRAs, and then treatment was interrupted to see whether the cure has happened. Because if the cure has happened, then there shouldn't be any viral rebound. And these trials have all failed. And we think it's because we haven't looked close enough. So what we would like to do now is to study in much more detail the different steps being required to make cells immunologically visible, but also where are some blocks or barriers which prevent the cells to go into full-blown reactivation mode. And actually, we found a lot of surprises, and this is very exciting work, which is still in the pipeline, still not published, because we want to make it perfect. So Julia showed that some latency reverting drugs actually facilitate HIV-1 reactivation. I mean, this is what they are done for. But if you look closely, they also interfere with some steps, meaning that the cells get more resistant to killing. So they express a lot of viral stuff, but the cells just don't die because these drugs actually make cells more resistant to being killed. And Dylan and our team is looking at viral contribution, which even further complicate this problem. So we have wonderful LRAs, which make all these things perfectly well, but one of these HIV-1 proteins makes the cell very rigid and resistant to apoptosis, which is killing. And we think we have to hit this one as well. So it's very exciting work. We believe it could make a difference in the field of HIV-1 cure, at least to fill a gap between what we know and what is needed to really achieve this goal. I think I finished with the scientific part, and I will now share some unsolicited advice and, of course, the obligatory childhood pictures. 4 advices: aim high. If you're comfortable, it's time to move on. Are you terrified of what's next? You're on the right track. I think you will believe me that this has been my strategy so far. I'm not sure if I will be able to continue this, but so far it will. Think positive. This sounds so trivial. Sometimes it's really hard work to think positive. So it's not meant lightheartedly. We all experience failures, either in scientific aspect where grants are not selected, papers get rejected, we get very nasty reviews sometimes. I didn't show all of this today, but this happens to all of us. So it takes active energy to think positive, to believe in good outcomes. And to me, it has given me strength to advance. But it's not easy. Think twice. I think people will really recognize me not only in this picture, but also in this quote, which I think could really come from me. Don't accept this won't work. If you believe, yes, we can, it's possible. And sometimes we ourselves tell us this is not going to work. And in a second or third thought, we can make so much things possible, which we deemed impossible in the first instance. And I also mentioned that I take my time to publish. I like to be rigorous and complete. So resist the pressure to publish fast at the expense of completeness and rigor. So fast science is a bit like fast food, some people say, and I think maybe I find myself. At the very end, I would like to thank my family for making my life colorful and spicy, and for all the support I obtained, and would like to close here 1 minute early. And I'm happy to take questions. I'll open it up now to online and I'll pick up on you. The shocking kill, I kind of think is that where what's the product good? I where how it was happen. As we go down, but actually that way that you yes, in the context of HIV/AIDS, many clinical trials with different LRAs have been conducted, and these were combinations which were tolerated. The clinical trials did not fail because of toxicity or stuff like this. So they really saw an increase in the viremia, which is what you expect if you reactivate the virus. You would like to see increased RNA levels, but the virus reservoir did not shrink, which really means that the kill part of shock and kill didn't really work. If you're asking about other pathogens, I mean, as a virologist, I could think that it would make a lot of sense in the field of herpes viruses because they also have not an identical, but also a persisting strategy where you would like to get rid of cells being positive for the viral DNA. I think there's not so much effort being put into herpes virus infections because maybe the need is not being recognized as much. TB, I think there's some analogies where I think people have thought about similar concepts, but of course this would need to be approached very differently. The other problems we have with the shock and kill approach is that the therapy is so good. People are, if they are well supported medically, they don't suffer much side effects and they can have a good life. So to convince them to go into shock and kill trials and then to accept, okay, we are going to stop your antiretroviral therapy to see if it works, of course, now comes at a more and more increased expense. So we have to balance these two things, I guess, which is an important step.
Okay, thank you very much. So I was going to ask, doing virus entry for specifically HIV-1, since the CD4 cells is not sufficient for its entry. So how does it have the preference for using either chemokine receptor 4 or 5?
Yeah, CCR5 versus CXCR4. So interestingly, in the context of transmission from one person to the other, 99.9% is being done by viruses which use CCR5 exclusively. So there must be a bottleneck in nature preventing CXCR4 using viruses to get transmitted. And this, I could talk more hours probably about this. So we don't really understand this, but there's a lot of hypothesis. So although a person living with HIV-1, especially at the end stage of the disease, can have a mixture of viruses in their body, so X4 versus R5 topic ones, or even viruses which can use both of them, only the CCR5 using virus is going to be transmitted. That's why we focus on that in the rat study.
Choice of two microphones, which is always a bad sign. Great talk, Christine. In terms of going back to shock and kill, what do you think might be the impact of immunologically privileged or sanctuary sites like the brain or the genital tract. Would that make it harder or?
Absolutely. I mean, we would be delighted to see a reduced reservoir looking at peripheral blood or some lymph node biopsies, which has not been achieved yet. Yes, the hard work will come when we have to reach, as you mentioned, like the brain, which is very difficult to access by many pharmacological compounds. On the other hand, we have to distinguish between functional cure and sterilizing cure. So I think there's a general acceptance in the field that maybe we do not need to kill every last HIV-1 positive cells, but it will be enough to reduce the size of the reservoir to a certain extent, and then the immune system will kick in and do the rest, which is functional cure. So you would still find HIV-1 DNA, but the person would be aviremic. It would control in the absence of antiretroviral treatment. But yes, absolutely, and we are far away from thinking about these problems because we are still working on the very initial idea to shrink the viral reservoir meaningfully.
Thanks, Christine. So for the first time ever, we now have people living to quite an elderly age with HIV. Do you think that the field of HIV is going to kind of change over the next 10, 20 years as we start having geriatric populations who've lived with HIV? And what do you think that field is going to look like?
That's absolutely correct. So, homes for elderly people are more and more experiencing the problem of people 70 years, 80 years being HIV positive, and they experience totally new issues which we don't know and which need to be studied. So absolutely, this is already an ongoing topic and probably requires more research. I would say when we attended the HIV Cure conference in Durban a couple of months ago, because of these political developments, now the field is really very keen to intensify vaccine and cure research. Because as I told you in my introduction, there was also a sense in the field that, yeah, we will somehow get it done with treatment, which I think now is proven wrong, very sadly. So I think the newest development is the field is really to focus on vaccine and cure. And in addition, to focus this research in affected countries. So it has been very much dominated by the Western world doing these studies. All these cure studies have been done in young, white, male Americans, which does not reflect the situation we find in South Africa, where women are infected with other subtypes and are exposed to very different conditions. So there's a very huge, I think, motivation to now really switch gears to not centralized, but really that the research should be focused in Africa, involving African researchers and the conditions which prevail there. And I think LFDM has a good chance actually to be part of this by establishing good relationships with key people in Durban, for example, or Malawi.
Make this the final question. So it's striking with antiretroviral therapy that doesn't appear to be development of resistance. So that, to me, is extraordinary, considering other viral infections or other infectious diseases. So what is the basis for the secret of success in the HIV field that other infectious disease pharmacologists can take learnings from?
Well, I think 2 points. First of all, we have a tremendous number of different molecules which can be combined in the context of antiretroviral therapy, which is not the case for many other viral infections. So the chance of development of resistance is lower because, or if they happen, we can switch to another regimen and then you get rid of the problem. However, the notion that resistance does not occur or is a rare event is also not correct. If you look in depth, you see definitely resistance conferring mutations arising. It's again a testing issue, I guess. And I personally would not want to predict that this will never become a really serious issue. I mean, we have other pathogens where we also thought we are very safe, we have great drugs, and this won't be a problem. And even if HIV-1/AIDS has a whole portfolio of drugs, at some point they may not suffice. So I'm actually a big advocate to keep going on identifying new molecules, new mechanisms, because we might need them. But again, I think it's not that there is no resistance problems. They happen, but they can be then circumvented by monitoring. So if they're identified early enough, then people can switch to other regimen. And yeah, in other parts of the world, these resistance happen, but we do not necessarily identify because we don't have them.
You definitely could. It's really one out of ignorance, and that is that there's a lot of interest in gene editing approaches, for example, knocking out CCR5 to effect a functional cure. Where is that these days, and what do you think it will do in terms of democratizing access to powerful HIV approaches?
I think the gene editing approaches are not advanced enough to apply them at large scale in patients. There are different approaches where you can genetically modify and try to get rid of CCR5. I think that the strategy works has been shown in animal models in principle, but I think the barriers to use enzymes which cut and paste in our genome with potential off-target effect is really not there yet. So I personally don't think this will be the golden solution to HIV-1 cure, but maybe I'm wrong.
Okay, I think we'll conclude there. I don't think there's anything online. The series pauses for the summer, and we start again in September. We're still on the radio. 17th of September, and please visit the website. You'll have a look at the meetings thereafter. Please join us in a moment for drink, nibbles. But if we can just give one final thanks to Christine for the